All right, well, great. Thank you very much for the introduction, as well as, to you and the entire committee for the invitation to give a seminar today. And I'd really just also like, to say thank you more broadly for the really great seminar series that you guys have been running for, months, maybe going on, a year now. You've had an incredible lineup of speakers, and I know people are enjoying tuning in every week. So as was just introduced, you can see right here in the title slide, my lab really focuses on the deubiquitinating enzymes. So for those of you that are regular attendees to this seminar series, this will be your, second dose of DUBs. David Commander gave a really nice talk on the enzyme family a few weeks ago. But I think that you'll find my talk to be very complementary. While he focused more on, DUB regulation and understanding and investigating the emerging roles in biology and disease relevance, I'm going to be more focused on, the chemistry and developing chemical probes and the strategies that really are working best for us to achieve these first-in-class inhibitors. So here are my disclosures, which you can read. I would just like to, take a moment to acknowledge on this slide that much of the work that I'm going to be talking about today, in particular, chemoproteomics experiments, have been done in collaboration with Jared Marto, who's a fellow faculty member here at Dana-Farber. So here's just a brief outline for the talk today. I don't think DUBs need much of an introduction, so I will keep it brief, but the few points that I'll want to touch on are some of their roles in disease, give you a little bit of a brief history of how the DUB field's really evolved over the last 20 years, and then really highlight how there's been, really nice progress recently in utilizing chemical probes to really advance the DUB field, and in particular, addressing questions of DUB substrates, DUB functions, substrate scope, and redundancy. Some of these key questions really that people have had for many years. And then this will, lead into, our real motivation to pursue a target class approach to start to really accelerate the pace at which chemical probes are being utilized to really break the field open. And in particular, I'll talk about some of our efforts in library versus library screening. So by that, what I really mean is some of the developing DUB targeted libraries and screening them against libraries of DUBs, which really opens up a lot of new opportunities, both for individual DUBs developing probes, but also learning some general design principles that we can then, really leverage to even further expand the number of DUBs that we can target. And then I'll talk a little bit about how we're thinking, about which DUBs to focus on now that we have a growing list of members of the family with inhibitors. And this is going to include some work in chemical genetics, as well as, integrating multi-omics data, both data we generate and data that's already in the public domain. So let's see, how can I get this? I have all my titles covered up. I guess I'll just leave it that way and work with it. So DUBs, as everyone knows, are a family of proteases that cleave ubiquitin from substrate proteins. They thus play, a critical role, not only in protein stability, but also in regulating, non-degradative ubiquitin functions. And thus they're broadly implicated in nearly all, physiological and pathophysiological processes. Working at Dana-Farber, I think a lot about their roles, in cancer. And, what's really, I think, striking is how broad it is, both in terms of mechanism as well as tumor type. And so, one class of potential drug targets are those DUBs that have gain-of-function oncogenic mutations. I've highlighted a couple of those on this slide. One being, USP activating mutations that are found in pituitary adenomas. And a second, example that's been studied quite a bit are USP6 rearrangements that occur in a number of different bone and soft tissue neoplasms. Another kind of, class of DUB targets that people think about, or another way to think about them, are those that reside as part of protein complexes. And so, for example, there have there's been quite a bit of interest in developing small molecules that target the DUBs that are bound to the proteasome and have roles in cleaving ubiquitin before substrates are degraded. And these really have been pursued, largely in the context of multiple myeloma that has become refractory to proteasome inhibitors. But really, probably the most attention really is paid to the DUBs that are stabilizing particular disease-relevant proteins, really with the notion you inhibit the DUB, you promote degradation of this disease protein. So I'm highlighting just one of the most well-characterized examples for you here, USP7 stabilizing MDM2, which then has downstream effects on tumor suppressor P53. But really, there are a large number of, DUB disease protein pairs that have been reported. And, a really exciting, I think, avenue is being able to go after some of those where the potential substrate has proven to be recalcitrant to efforts to target it directly. And so just to, give a little bit of a history of how the field has evolved, it was probably about 20 years ago that DUBs really started to grab the attention of researchers. It was during this, period in the early 2000s where there were several high impact papers that were published that really nominated DUBs as targets in a couple different tumor types. And this was the exact same time that the first DUB inhibitor, high throughput screen and then inhibitor was reported. Following, some of these early studies was a, period over the next 10 or 15 years that I usually view as having quite a bit of ups and downs. On the inhibitor side, which I'm showing on kind of the top half of this timeline here, there really were quite a few new inhibitors that were discovered, as well as development of new reagents for biochemistry and activity-based protein profiling. However, we later came to realize that many of these early generation inhibitors that were undercharacterized, and they were undercharacterized because we didn't yet have nice panels for assessing selectivity, were in fact multi-targeted pan inhibitors. And unfortunately, this had led to a bit of a muddying of the literature where these had been used to assign novel DUB function. In parallel to efforts in the inhibitor space were a number of studies that, to understand DUB function and role in disease. These span sequencing studies, large-scale genetic and profiling studies, and these really nominated DUBs, as drug targets. Many of these were very exciting, opened up many exciting possibilities. But, in some cases, we also were seeing a number of conflicting reports, really leading to some uncertainty about what really were going to be the best areas to focus on in thinking about prioritizing DUBs for drug discovery efforts. And so while, I refer to this period of having, some ups and downs, there really were a lot of important, advances, and it really, laid the groundwork for what's been a really exciting last several years in the DUB space, where we've really started to see a small burst at least, of potent and selective inhibitors, and then nice demonstration of how these pharmacological tools really can be utilized in both biased and unbiased studies to advance the DUB field. And so over the next several slides, I will just show you briefly, a couple of these examples and really highlight some of the important, learnings more generally from them. Excuse me. So the first point really being that, potent and selective covalent, as well as covalent active site, as well as non-active site inhibitors are achievable for DUBs. This may sound like a statement that, kind of goes without saying, but it really was a question, 5 years ago in the field, are we going to be able to get potent and selective inhibitors for a wide range of DUBs? And, it was maybe not such a crazy question when you keep in mind that for other families of cysteine proteases, the inhibitors that had been developed, they were targeting covalently the active site, but they were achieving selectivity because of different, substrates. The substrates looked very different, whereas for DUBs now, they all share ubiquitin as a substrate. So what features are we going to leverage to get selective inhibitors? Unfortunately, we now see from work on several different DUBs getting these selective inhibitors is very achievable. I'm just showing you data here for our second generation USP7 inhibitor. We developed this using structure-guided approaches. This is a irreversible inhibitor. It's very potent down in the picomolar range. And importantly, we've been able to show that it has a very high degree of selectivity. And we've shown this across multiple assay platforms that focus on purified DUBs, native DUBs, as well as the entire proteome. As you may be aware, USP7 has certainly been one of the most focused upon DUBs for inhibitor efforts in recent years. And so, what I find really exciting about those efforts kind of collectively is how they've revealed a number of binding pockets on DUBs that one can really think about leveraging for their future programs. So of course, there's the catalytic site, which we are targeting in the example I just showed you. You also can achieve non-covalent inhibitors that bind kind of what's referred to as the tunnel or the S3 to S5 site. But there also is, at least, one, and I suspect more, distal pockets that you can target with novel inhibitors. So just the second point that I wanted to make as part of this introduction was that, these chemical probes really are turning out to be quite powerful and unbiased studies. And in particular in proteomics, where, you really want this temporal control. So for our USP7 program, we ultimately really prioritize doing these unbiased studies to start to generate hypotheses for, key functions of USP7. And the truth is that we moved really to this focusing on these unbiased studies after we, spent a fair bit of time struggling with validating some of the wide range of different substrates that had been reported in the literature. And what I found, really quite striking about our work with these unbiased studies is how they really converged on one pathway. And the one pathway they converged on was MDM2-P53, which was the first reported, pathway to be regulated by USP7. But just briefly to take you through the data, starting on the left is the quantitative proteomics, where we're looking at changes in abundance. This was work done in collaboration with Eric Fisher's group here at Dana-Farber. But what you can see is that there's a decrease in MDM2, increase in P53. But more broadly, I think really the important thing to note here is that there are not a tremendous number of proteins that are changing in abundance. And so certainly an open question and a concerning question for the field has been, what is the substrate scope of DUBs? How many things are changing when you inhibit a DUB? How is this going to impact, toxicology? So this is, one example, one context, but, I find it very encouraging that substrate scope may be, more limited than people had previously been assuming. And then in a couple other studies, we did on transcriptomics, this further highlighted really, P53 gene signature. And then, a nice study in the middle is profiling against cancer cell lines, which then starts to link a role to P53 to phenotype, with, the phenotype here being growth suppression, profile the compound against a large panel of cancer cell lines. And then the profile was compared to genome-wide CRISPR knockouts that had been done across the same set of cell lines. We see that the profile of our compound is most similar to knockout of USP7, telling us it's on target. Furthermore, the profile is highly similar to knockout of MDM2, anti-correlated with P53 knockout. And further, we can see that a mutation in P53 predicts for insensitivity to USP7. Really driving home the point that, at least generally across multiple lineages, MDM2-P53 is going to be an important predictor of response. But this then also leads me to just the third point I wanted to make here in terms of, using small molecule chemical probes to understand DUB function. And that's that, these unbiased studies will have their limitations, and that for the most part, they are being done in a single context or with a single fitness metric. And so while we, in collaboration with Kim Stegmeyer at Dana-Farber, were ewere able to confirm that in Ewing sarcoma, growth suppressive effects of USP7 inhibitors is driven through a p53 through the p53 axis. There have been a number of nice studies from other groups. I'm highlighting just one on the right side of the slide here that really credential a role for USP7 in mutant p53 settings. The one that I'm highlighting here is a dependency for USP7 in small cell lung cancer that has mutated p53. This has been shown both in cell culture as well as xenograft studies. And the mechanistic investigations that have been done really point to USP7 impacting cell growth through p53 independent DNA repair pathways. And so, just to briefly kind of, recap, some of the lessons here, not only from USP7 that I've shown you, but also from some other nice studies that have been done with inhibitors of USP9X recently, work from, Michael Clegg's group in collaboration with Forma, as well as some CSN5 inhibitors from Novartis. I think it's very compelling now that potent and selective covalent and non-covalent inhibitors can be achieved. We really, the field certainly needs combination of focused and unbiased studies to elucidate function. And it's very encouraging to see that, as we're starting to do more unbiased proteomics, substrate scope, may be relatively modest. We are not seeing redundancy being a major factor thus far. But certainly, we need to do further exploration, in more context. But to me, what this all really supports is that, we really need to get more small molecule inhibitors, move beyond the, 3 to 5 that most of the field has been focused on, and try to get small molecule probes for a wide range of different DUBs, including some that are very understudied, so that we can start to advance not only, those individual DUBs, but start to really learn generalities for the DUB field. And, I really envisioned us being able to accelerate by taking this, target class approach. This target class approach, it's been very successful for other enzyme families. I think most notably, kinases, where these libraries of ATP mimetics have been extremely powerful. And you can draw a number of parallels between kinases and DUBs. Like kinases, DUBs are a large family of structurally and functionally related related enzymes. In total, there are about 100 that's split into 7 families, with 6 of the 7 being cysteine proteases. In terms of structure, we know that all the cysteine protease DUBs have conserved active sites, characterized by the catalytic cysteine that's stabilized by a couple nearby amino acids. And then further from these structural studies, we know that DUBs within a family adopt a common fold, but with limited sequence homology. And then as you start to look across these different, DUB families, you really do see divergent architectures, which I think, really adds up to being able to leverage some of the, common and distinct features to be able to rapidly develop, probes across the enzyme family. And this is really what I envisioned our approach to look like. We would have DUB-focused small molecule libraries that we would profile for activity and selectivity across ideally the entire target class, to then have nice, annotated libraries. And then with these annotated libraries, we'd have a number of opportunities arise. We could pick specific DUB-ligand pairs for medchem programs to get probes. Then we could use these probes to test hypotheses and generate hypotheses. But even before generating, doing the medchem to generate any probes, we could take these entire libraries and utilize them in phenotypic screens to start to identify, novel function, and then link that function to specific DUBs or, perhaps even subsets of DUBs. And, going into this, thinking about our ability to test hypotheses, there was a little bit of concern. Just DUBs are an emerging family of enzymes. They're relatively understudied. Here I'm just showing you, publication count for the various DUBs. And while, it's quite high for a few, it's quite low for many. which means that we don't really have strong hypotheses of function for, many of these DUBs, especially when there's only 3 to 5 papers and they each suggest something different. And that was what, really motivated us, and I'll talk more about this towards the end of the talk, to think about how we could, do some of our own, CRISPR screening to identify function, which we've done through, knockouts followed by RNA-seq, and then really integrating this with the really great, work that's been done, by other groups to look proteome-wide at the function of different function of proteins, so that we could then integrate this, with the literature to generate some higher confidence hypotheses to test. And so, when the lab was starting, I don't know, 5, 6 years ago, we would have loved to jump right in with DUB-focused small molecule libraries. But of course, the problem was we really had no idea what one would look like. But we did have criteria in mind for, the types of compounds, the types of scaffolds to look out for as, we were doing work on individual targets, as were others in the field. And so really the compound profile that we were looking for inspiration was, having a scaffold with at least one compound that inhibited its primary DUB target with an IC50 less than 1 micromolar. For our first generation library, we thought that going for covalent targeting the active cysteine would probably be a smart way to go, since it is the most highly conserved feature of DUB. So we wanted, this scaffold to covalently label the catalytic cysteine. Thinking about library generation, we needed to synthesize it using a modular scheme. And then, preferentially, this scaffold will be represented by multiple analogs that exhibit, disparate DUB selectivity patterns. And specifically, we would like examples where we see multi-targeted behavior with one member of this scaffold, while, there'd also be highly selective examples for a single DUB with other examples. And fortunately, compounds meeting these criteria have emerged in recent years. I won't discuss these in depth, but just to highlight, there have been the N-cyanoperyllidines, which have been studied by Mission Therapeutics and others, where there are nice examples that are selective for UCHL1, but there also are examples that are multi-targeted, targeting not only members of the UCH family, but other families of deubiquitinating enzymes as well. And then for USP7, we've had these tetrahydroacridines, so a bit of a, a unique warhead here, but a warhead that's been featured in USP7 selective inhibitors, as well as multi-targeted examples. And then finally, we have this, alpha chloric ketone based example, which is multi-targeted towards a number of different enzymes within the USP family. But as we start to modify this, benzyl group into, larger groups or adding substituents, we start to see that we can change the spectrum of targets. So we really then leveraged some of the features that we observe in these compounds, along with some DUB-ligand and DUB-ubiquitin co-structures, to design our first generation DUB-targeted covalent active site library. And so there's kind of a lot to absorb on this slide here, where we're trying to capture our overall design strategy. But essentially, we wanted to leverage 3 key features of DUBs. First, of course, being the the active site, covalently targeting the catalytic cysteine. And so we wanted to there, employ a number of different electrophilic warheads, really taking a lesson from the, inhibitors I showed you on the previous slide, where we were attaching these electrophiles kind of directly to a ring. It was kind of a defining, we found that to be a defining feature of the examples shown on the previous slide. We then wanted to really leverage or, take into consideration the region approaching the active site, which would normally be bound by kind of a gly. And so it's a narrow channel. And, we figured we would need to fill this narrow channel potentially with alkyl chains. Maybe there'd be space for some small heteroaromatics. And in some cases, maybe we could even get away with really short linkers. And then as we started to, move further away, have these compounds move further away from the active site, We anticipated that they would be forming interactions with blocking loop, 1 and 2, which differ quite a lot in terms of, sequence as well as positioning across DUBs. And we thought would probably be an excellent region of the molecule, or I'm sorry, of the DUB to leverage to start to, get unique interactions and selectivity. And so really what we ultimately did were, combinatorially assemble these non-covalent building blocks, the linkers, and the electrophilic warheads, really hoping that it was going to be specific combinations of each that would be driving targeted DUBs for each of the individual compounds. And here are just some representative library compounds. I think these just nicely highlight, again, as I mentioned on the previous slide, a defining feature of the library, which is where we have the reactive component, in most cases, not all, really being defined as a combination of ring system attached to electrophile. And then I just wanted to note that our initial library is about 200 compounds, but what I wanted to note here is the average molecular weight's about 350. So they're bigger than fragments, but they're not necessarily fully functionalized compounds. And so we really do kind of handle them as somewhere in between in terms of the concentrations that we use for screening, as well as thinking about how do we call actives and what is a respectable starting point for a compound that comes out of this library. So with the library in hand, we needed to pick an assay to utilize to screen the library. And the assay platform that we selected was competitive activity-based protein profiling. So this is shown just kind of in the schematic here. I'll just walk you through it briefly. But essentially, we will take, cell lysates, or this also can be done with live cells, treat with an inhibitor, which will bind some fraction of DUBs within the cell. We then can co-incubate with an activity-based probe, which will label any DUBs that are not, blocked by the inhibitor. Then do an infinity enrichment step, quantitative LCM MS. And really what our readout then is for, all the various DUBs and all the various compounds, to what extent did each compound block labeling of each DUB by the activity-based probe, therefore inferring a binding event, and since it should be targeting the catalytic cysteine, an inhibitory binding event. And the reason we really focused on this assay was because it gave us potential for broad coverage of the DUBome, as these activity-based probes can react quite broadly. I also think very importantly, it would allow us to find hits for many DUBs, which was of course our overall goal. And then this would allow for us to pick compounds to follow up on based on selectivity, perhaps even more as a higher priority than potency. And then further, we'd be able to have SAR trends not only for the individual DUBs, but as well as the entire target class. And so this is the assay we use to screen the library. So across all runs, we did about 25 runs to screen the entire library. We detected 65 DUBs total. And it was about 56 that, we consistently detected in every experiment. And these 56 really span 6 cysteine protease DUB families. So we were hitting all of them. And here are the results from the screen. The heat map here is showing you darker red, the greater extent that the compound blocked labeling by the activity-based probe, all the compounds along the top, DUBs, DUBs along the side here. And so just looking at this heat map qualitatively, you can see we had active compounds first and foremost, which was certainly a relief for a first generation library. But you also can see that there's quite a bit of variability in terms of the target spectrum, of these compounds. And it looks like we're getting pretty broad DUB coverage. So these were, the first two things that we really tried to quantitate. And so in terms of active compounds, we defined these as greater than 50% blocking of ABP. And based on this definition, we saw that just over half of our compounds were active, targeting at least one DUB. And so we were really, pleased by that number. But I think what, really got me more excited was when we kind of drilled down into how many DUBs are actually targeted by each of these compounds. If this is, a bunch of multi-targeted compounds, it's maybe less exciting. But what we saw was that we had quite a large number of compounds that were targeting only, 1, 2, or 3 DUBs. And I'll show you the data for these compounds specifically, on some subsequent slides. So a large number of active compounds and indication that quite a few of them were exhibiting good selectivity. In terms of DUBs targeted, we saw that, about 2/3 of the total DUBs that we detected were targeted by at least one compound. And further, that half of those were targeted by selective compounds. And so in the bar graph, for the USP family, for example, we see that among all compounds, 28 out of 42 were targeted. And then when we restrict this just to the compounds that are hitting 3 or fewer targets, we see 17. So when we consider all compounds, we've hit members within 5 DUB families, although this does drop when we focus in just on the most selective compounds. So I want to come back then to, what are highlighted here is the most selective compounds targeting 1, 2, or 3 DUBs. This work was done by Adrian Chan, who's a joint graduate student between myself and Jared Marto. And when he showed me this pie chart, my first gut reaction was this almost sounds too good to be true. So I asked him to, show me the heat map for just those compounds. And when I saw it, it actually looked pretty promising. We are seeing that many of these compounds do appear to be pretty potent and pretty selective. We're not just picking out a handful of compounds that have target a few DUBs just over 50% and a bunch more just under 50%. But when I really started to get excited about the output from this screen was when we started looking at the structures, for each of these hits, and we started to observe how strikingly similar hits for individual DUBs were. And so I'll just kind of show you a few examples on this slide, starting with UCHL1. Where we really saw that nearly all of these examples here that had good selectivity for UCHL1 had similar chemical features across all 3 components of the compound, the electrophile, the linker, as well as the non-covalent building block. All of the electrophiles, they contained these cyanamides, and they were all within alkyl rings. For the linker, it had to be a heteroaromatic, and by comparing the active compounds to inactive compounds, we could see a strict requirement for a hydrogen bond acceptor here at this position that I'm highlighting. And then in terms of the non-covalent building block, only those that contained aromatic rings really were active. It was a very similar story for USP3 in that we saw compounds were very similar. It crossed all 3 different modules of the compound, but interestingly, very different looking from UCHL3. Very different electrophile, different linker, and now a different non-covalent building block. The trend continues as we look across other DUBs, just noting an interesting feature now for, these hits for USP28 that are all highly similar. This was one of the examples where we saw a pretty strict requirement for having a flexible alkyl linker between the electrophile as well as a building block. The final example I'll just highlight on this slide is USP48, and I just wanted to highlight this to really kind of capture electrophile diversity. Now for these examples, we're seeing alpha chloroacetamides, but interestingly, we're really seeing them attached to pretty bulky rings. And then, this was a case where we had a very short linker and then, a small aromatic. And so individually, we really were able to pick out important design features as we were to think about optimizing compounds for individual DUBs. But I think bigger picture, these really, are compelling examples of the reality of, what we hope to see, which is that you really need a specific combination of warhead, linker, and non-covalent building block that are leveraging the catalytic site, the catalytic cysteine, the narrow channel where the C-terminus of ubiquitin binds, as well as the blocking loops that I noted to determine target spectrum. This is not being driven just by the electrophile. And I'll just highlight here a couple examples that I won't go into, but because I will talk about hits for these DUBs on subsequent slides. Nice inhibitor for USP9X and a couple, nice compounds with high degree of selectivity for the OTU family DUB, VCPIP1. And so really with, high confidence, in the data set, we, went in to do some more detailed analyses, really looking in one of the questions we wanted to address was substrate scope. There are a small number of DUBs that have been liganded. How are we doing in terms of expanding on that scope? I apologize for all of the sirens in the background here. And so, in total, we were targeting 45 DUBs. Based on, our analysis of the literature, we were, targeting 7 DUBs for which probes had already been reported, but 38 DUBs for which, no hits had been reported in the literature. And then if we restrict it just to, DUBs with selective hits, we have 23 previously unliganded DUBs that are hit by a selective compound. So really overall, this library versus library approach has enabled us to greatly expand the scope of liganded DUBs, providing a lot of new opportunities for developing inhibitors for individual DUBs of interest. And here just gives you a little bit more granularity on what those DUBs are. Here we are listing, showing the DUBs that were targeted by selective compounds. And we've also then further, in this complicated Venn diagram, broken down which DUBs are targeted by which electrophiles. And so you can see that, in some cases, we have DUBs with very strong preferences for a particular electrophile. And in other cases, we have DUBs that, react with a number of different electrophiles, but we can, still get selective inhibitors through, non-covalent interactions. And so I think, really at this point, having analyzed our primary screening data, we obviously had a high degree of confidence in it, but the question is, are they validating? And then can we take these and really turn them, quickly into probes? How is the progress going there? So we've implemented a number of different orthogonal assays to confirm and validate hits from our screen. I'm showing you here the example WH103C, which was one of our selective hits for VCPIP1 that I had kind of called out briefly in the heat map. And we were really interested in validating this in part because it was appeared to be one of the most potent and selective compounds in our screen. Yet there have been pretty much no, chemical probes developed for any members of the OTU family of DUBs. And so we validated this first just using our same ABPP assay, but now coupled with Western blot. We showed in a biochemical assay that it's inhibiting VCPIP1 with an IC50 of about 500 nanomolar. We used intact mass and MS analyses to show that this was singly labeling VCPIP1 and that it was in fact labeling as we expected the catalytic cysteine. We then profiled this compound against a panel of recombinant DUBs. This is a commercial panel available at Ubiquitin. And we saw nice selectivity for just this one target, VCPIP1. And then we followed this up with looking, using chemoproteomics to look proteome-wide. And here, all we really saw was that this compound was targeting 2 cysteines that are known to be pretty hyperreactive. I will note here that you do not actually see VCPIP1. It is not because VCPIP1 did not confirm in this assay. It's that, the peptide had gotten buried under a much larger abundance peptide. And so we're now doing some more targeted mass spec experiments to be able to, pull that peptide out and really quantify, the extent to which it is labeled by our compound when we're doing a proteome-wide experiment. So we then selected this validated inhibitor as our first test case for being able to rapidly optimize. And we were hoping we'd really be able to rapidly optimize because we have SAR trends, from the entire data set. And, also because we would really in our Assay, as part of our optimization program, be able to do parallel profiling against the purified enzyme of interest, as well as activity-based protein profilings. We're tracking selectivity the entire time. And it was pretty gratifying that it took about 50 compounds for us to be able to, drive down potency, quite a bit from 500 nanomolar down to 10 nanomolar, while retaining a high degree of selectivity as assessed against native DUBs using, again, the same chemoproteomics assay that we used in the primary screen, as well as against a panel of recombinant DUBs. This is, again, with profiling done at Ubiquitinate. So it is our hope, fingers crossed, I'm sure, for some DUBs it will be easier than others. But, this gives us confidence that given the data dense nature of our primary screen, we are able to rapidly develop probes. And really, kind of the other, an upshot of putting all of these medchem compounds through ABPP is that we also were able to open up some new unexpected opportunities. We started with a compound that looks very selective for VCPIP1. And then we started to see that for some of the analogs, we were eroding selectivity a little bit. We were picking up some other DUB targets. But by following some of the SAR trends, we predicted chemical features that might be driving interactions with particular DUBs, in particular UCHL3 and USP40. And so when we made compounds that incorporated those features, we were then able to get selective ligands for these two other DUBs. So through this effort, not only did we get a nice probe for VCPIP1, but now we also have starting points for a couple, additional DUBs. And in particular, in the case of USP40, we did not have any hits for that DUB in our original screen. And I'll just summarize the rest of our validation efforts. Thus far, we're getting a pretty high, rate of validation. Then this validation really has focused on the batch of most selective inhibitors, where we've had compounds that we've been able to confirm in at least 2 assays thus far against the DUBs that I'm showing you here. As you might expect for a field where it's still relatively early days, we've not been able to really pursue validation for all of these hits because we are reagent limited. But it's been a relatively small number of DUBs that we have not confirmed the ABPP results when we started doing orthogonal assays. There's 4, but I want to note for 2 of these, they didn't validate in biochemistry. We followed up with dose response activity-based protein profiling, and they confirmed there. So this might be an example where we see a real discrepancy between, native DUBs and, purified isolated DUBs. And this is a result we need to, follow up on further. And so, just to transition here a little bit from kind of the chemistry to some of the new biology we're able to do now. We screened these 200 DUB active site targeted covalent compounds across, 50 plus native DUBs. We were able to identify hits for quite a large number of DUBs, many of them by selective compounds. So this really greatly expands the number of liganded DUBs that are now candidates. Have starting points for medchem programs. We were really gratified to see that selectivity is driven by a combination of interactions with 3 regions of the DUBs, which really validates our design strategy. And we really demonstrated proof of concept in the case of VCPIP1 that potent and selective inhibitors can rapidly be achieved from these hits. So of course, there are now a number of next priorities. We're wanting to scale up our covalent active site libraries, and this is also going to require us to increase the throughput of our ABPP assay. This is in progress. I'm not going to talk about it today. We really have a push for prioritization of DUB-ligand pairs for optimization in biological studies. So I will touch on that briefly over the next few slides, as well as just, I think maybe in 1 or 2 slides, then touching upon ways we're thinking about increasing the diversity of our libraries, and that's through developing, finding scaffolds and making libraries or building libraries around these scaffolds that non-covalently target a wide range of different DUBs. So this I already showed you earlier, but it's just reminding you that, as we start to, find ourselves in a situation where we have a number of different DUBs that we could pick to work on, it's sometimes a challenge to decide which one. Because oftentimes either you have a well-studied DUB, but it's reported to do so many things you don't know where to focus, or you know very little about the DUB. And that's really, driven a really great project led by a graduate student, Laura Doherty, who's joined with Peter Sorger in the Laboratory for Systems Pharmacology, where we've been able to, integrate multi-omics data to reveal function and therapeutic potential of DUBs. And I'm not going to talk about this in great detail, but just the overview is that we did, DUB-wide CRISPR knockouts, followed by RNA-seq, really to be able to elucidate the transcriptional impact of DUB loss of function, pointing us towards, phenotypes or pathways. We then integrated this data set with cancer dependency map data, which, tells us, the DUB dependencies, but also reveals gene essentiality as well as codependencies. These all can be further integrated with connectivity maps, protein-protein interaction databases, proteomics that shows co-regulated proteins. What we can focus in on are codependencies with orthogonal support. So, we have multiple lines of evidence pointing towards a particular role of a DUB. And then oftentimes we can go back to the literature and see that indeed, there is agreement. We just didn't know which paper to focus on from the literature. Big picture, this analysis, and I think we're going to be submitting this paper here in just a couple of weeks, showed us that we have, revealed candidate essential pathways for 35 DUBs. We have candidate E3 partners for 23 DUBs. And we've been able to identify tumor types with increased sensitivity for about 34 DUBs. So these really then give us kind of a list that we'd like to think about prioritizing. Big picture, this effort has provided, some unexpected new insights for well-studied DUBs in particular, some potentially non-redundant functions for USP14 and UCHL5 that in particular are important, at least in the context of cell proliferation. It also has really helped us focus in on where we should be looking in terms of understudied DUBs, and I highlight just 2 examples here. One being this DUB USPL1. Actually, it's a desimylating enzyme, but characterized as a DUB. It had the second strongest phenotype in our transcriptomic studies, but there were only, which was striking to us because it hadn't been on our radar at all. And it hadn't been on our radar because there were only 3 papers on USPL1. But, through the results of the transcriptomics, integrating with the other data sets, it's pretty clear that where you need to be focusing for USPL1 is a role in mRNA processing, its association with a little elongation complex, and that there probably is therapeutic potential as a result of partial copy number loss in a number of different tumor types. And then for one of the DUBs we've of course been interested in, VCPIP1, this is another understudied DUB, but our analysis has really identified a network of proteins. We don't know how they all connect, but really for us to focus on to start to understand some of the relevant biology. And so the last thing I'll just touch on is really in addition to the multi-omics to identify individual DUBs to focus on, we've also been saying let's not pick DUBs or pick an individual DUB. Let's take our library, do phenotypic screens, and figure it out from there. And a lot of this work we've been doing focused in acute myeloid leukemia, in part because of a really great collaboration with Jim Griffin and Ellen Weisberg, who are experts in AML here at Dana-Farber, but also due to some unique attributes that make it an exciting area for us to focus. There were a lot of potential targets in AML. It's very heterogeneous. It's driven by usually a combination of two events. You need an aberrant transcription factor, such as one of the ones I've listed here, as well as a cooperating mutation in, one of a variety of different enzymes. Many of these remain, undrugged, or there are real limitations in terms of using the enzyme inhibitors in the clinic. But importantly, many of these have been shown to undergo ubiquitin-mediated degradation, but the DUBs involved in their regulation are pretty unexplored. So really the approach that we've taken, take our DUB-targeted library. We've started by screening just against an oncogene-driven line and a control cell lines. We're using the BAF3 system. This was kind of a test almost at the beginning of, can we use proliferation in these isogenic lines to pick out interesting compounds? And if we can, we then, would proceed to all of the downstream validation work that's required. And I have to say, using this, BAF3 screening system has been quite a bit more powerful than I might have predicted it would be. We found a number of compounds that selectively degraded mutant FLT3 over other oncogene-driven BAF3 lines, and importantly, was selective for mutant FLT3 over wild type. And we published this a couple years ago. More recently, and I think a very exciting example, given the real clinical need in mutant JAK2-driven neoplasms, we found compounds that selectively, suppress growth of BAF3 cells that are driven by mutant JAK2. They promote ubiquitin-mediated degradation. I'm obviously not showing you all of the data here. This is a paper that's currently under review, reporting this in detail. But we're selectively degrading the mutant JAK2 enzyme over wild type. And we see this not only with our small molecules, but also with shRNA or CRISPR knockout of the relevant DUB target. So we have a number of, emerging stories like these. Having our library is, really enabling these types of chemical genetic screens. And of course, we're using a number of different phenotypic assays, but at least in the case of AML, we're getting a lot of nice mileage out of these BAF3 screens. And so I will just try to rapidly conclude here by saying that, to start to gain access to even more DUBs, one of our focuses is now targeting these other libraries. It's taken a lot of high throughput screening to start to identify the scaffolds that we're going to be utilizing to build out targeting these pockets, as well as structural biology work, but we are finally now there. I will skip over the details of the screening so that I can really just, wrap up and say that, DUBs really are an emergent target class. I think there's tremendous therapeutic potential. As therapeutic targets, as well as, being able to target these, thinking on the chemistry side, targeting at multiple different binding sites. My lab's really been focused on a platform for DUBs that we, have differentiated ourselves by having these DUB targeted libraries, chemoproteomic screening platforms, and taking diverse and, approaches to interrogating the functional proteome. So I think the next decade is going to be a really exciting time for DUBs. It's been tremendous fun working at Dana-Farber, building the lab with a great group of students and postdocs, collaborators, and of course, I'm very grateful for funding. And I couldn't quite see the time here, so hopefully there's still at least a little bit of time for questions. Thank you, everybody that has tuned in.